interesting. And what I'm going to talk to you about today is related to epigenetic switchings and how these can use the noise to adapt to fluctuating environments. I think this is probably the only crowd ever I have spoken to that I don't have to convince you of any of this. But I still wanted to mention really quickly, like I understand biological noise as intrinsic to biological processes, and as such, it is omnipresent. It can also have very clear functional activities or presence. It is also very informative. We can use it to understand better or to gain information about the state of the process in any given moment. And finally, it is also evolving. It is both being shaped by natural selection and as well affecting the way evolutionary dynamics go. And I think the example I'm going to present today very much goes into these 2 points here. But before jumping into the topic, I wanted to take a minute to explain a little bit the general view of my research group. We are called the Evolutionary Systems Biology Lab, and as systems biologists, we very much care about understanding how gene regulatory circuits work. And by adding this evolutionary perspective into the mix, we get to ask as well the why of the specific form of these systems. I believe that to be able to answer this question, we also need to take into account both the omnipresence of the biochemical noise, as well as understanding how natural selection actually shapes this system. And in turn, this will also help us to understand the role of biochemical noise, as well as deducing what are the evolutionary trade-offs that are directing the evolutionary dynamics at the end. I think that together, this will allow us to get some insight into the structural principles that are shaping these gene regulatory circuits, and as well the very much needed mechanistic understanding for evolution. And I know this is a little aspirational. That is what we believe in the group. So now going into the topic, I'm going to be talking about epigenetic systems. And we understand epigenetic systems in this very, very general abstract way. It's any system that allows you to have for just one genotype in a given environment to present 2 or more distinct phenotypes, right? And this is actually possible thanks to having a dynamic reinforcing feedback into the system. And a good analogy to think about this is precisely the phenotypic landscape. It's a little more general than the Waddington landscape, but inspired by the same idea. And these phenotypic landscapes for these epigenetic systems will be characterized by having 2 different valleys or attractors, each of them corresponding to the phenotypes that we are interested in. Now, I wanted to highlight here that this phenotypic landscape very much will depend on both the genotype and the environment. I'm going to make a little references to previous talks. I think what Francesca is suggesting is incorporating the dynamic part of how the actual state of the system will affect the phenotypic landscape, because it is affecting both the environment and potentially also kind of the genetic background. But anyway, for us here, there are 2 important properties about having this type of systems. The first one is that it can show memory. In other words, in which the 2 possible attractors your system is in a particular moment will very much depend on the previous state. So it's in a way remembering what was happening. Now, if we add noise into the mix, then a stochastic switching between these 2 different phenotypes can appear, and that allows for heterogeneity to arise or to appear in an otherwise homogeneous population. And that is what we are interested on as a function. Because if we think about fluctuating environments, that is a reality all organisms have to deal with, then having this heterogeneity in the population can be advantageous. These epigenetic switches then will allow you to have true memory, this epigenetic inheritance, and through the stochastic transitions, the phenomenon of deadheading, which basically means that a population tries to anticipate environmental changes by introducing this heterogeneity in the population, sacrificing a few individuals having the non-optimal phenotype in the current environment, but being prepared for the potential change later. We wanted to understand, to explore what will be the evolutionary conditions that will allow for these systems to arrive. What is the selective advantage of epigenetic switches? For that, without getting into many technical details, because I don't have the time to, I am kind of regretting putting this slide here, but anyway, we are going to be thinking about the simplest gene regulatory circuit that can present this epigenetic switching. That is a self-induced gene. And what I really wanted you to take from this slide is that basically, if the environment is fluctuating between 2 different states, then there are 2 different strategies to deal with this. One will be just genetic adaptation. Every time the environment changes, the population has to migrate through mutation in the genotypic landscape until it finds it finds a solution that is optimal from one environment or the other. And then we have this alternative strategy that is epigenetic switching, where the phenotype is actually bistable already. It has the two phenotypes consistent, and we have a stochastic switching between them two. I also want you to take from this slide that anyway, doesn't matter which strategy you are doing, you have to deal with the biochemical noise, right? Now, to explore what are the evolutionary conditions that favor this, we are going to be doing in silico evolutionary experiments. Our model is actually very simple, but it already has many components, many things that can happen that makes it interesting. But we are going to be assuming that the environment can only be in two discrete states. Each state will be characterized by having a different optimal expression level or protein level. And we are going to be assuming that the frequency to change from one environment to the other is fixed. Now, that can be relaxed, and the plots or the results that I'm going to show you today don't go into that, but they very much hold even if we introduce noise into the environmental fluctuation. Now, for each generation, we are going to be assuming that we have a fixed size population. Each individual has a copy of the gene circuit of interest, but with its own genotype. Given the genotype, we can simulate the gene dynamics. We do this taking into account the intrinsic noise, the gene expression noise of the system, and given the phenotype, the expression level that we observe, then we can assign a fitness scores to each of the individuals. Once we have the distribution of fitness in the population, then we can select the individuals to be cloned, allowing for some random mutations to appear. And this is a generation cycle that we keep repeating through time. Now, in blue boxes, you can see the parameters, the evolutionary parameters that we are playing with, that we are exploring. And I wanted to highlight here that basically we have several sources of noise already. We have the environmental fluctuation noise. We have then when the mutations occur and the form of the mutation is random. And we are also allowing for some stochasticity in the selection process. But the most important one for us here is the gene expression stochasticity, because that will be the only source that can drive the stochastic switching in the epigenetic switches that we are interested. For that reason, I don't know why I go double. We are also running these control experiments, just removing that noise from the mix, assuming that the gene expression is deterministic, and that is going to be important to make some of the conclusions later. Okay, so I don't have much time. I think it's a really beautiful story. I hope if you are interested, you either approach and discuss it with me or look at the paper. But there are 5 points that I want to make really quickly during these few minutes. The first one is that we observe that the populations adapt to the fluctuating environment per se. For that, here I'm showing you that from an arbitrary initial condition, an arbitrary genotype, that is actually adapted to one of the environments, but not the other, the population is going to start moving, exploring the parameter space, the genotype space, until it finds a good solution for the fluctuating environment. This actually initially decreases the fitness of the population, but it doesn't matter because the population doesn't have a knowledge that the environment is going to keep changing. It still tries to adapt every time the environment changes. But eventually, it finds a solution that is adapted to the fluctuating environment per se. It allows it to adapt efficiently every time the environment changes. Now, to be able to explain this, we take advantage that we actually have the full information about the system. Is the good thing about doing mathematical modeling and in silico experiments, that we can actually go back and look at the details, right? So for our model, we can actually build this genetic potential map. It is telling us the distance between the optimal solutions in terms of the genotypic space, the optimal solutions for each of the environment. So what we are observing here is that the population originally migrates through the gradient of the genetic potential map, trying to maximize this. In other words, to be able to adapt more efficiently every time the environment changes. And it results that is precisely where the genetic potential is maximal, where we have our bistable region, where epigenetic switching is possible. This is not that surprising because in an epigenetic switching, you already have the two phenotypes without requiring any mutation. In other words, we have a zero distance between the solutions. And what you can see here is that this area here is showing the bistable solutions that match the low environment, the bistable solutions that match the high environment and one of their phenotypes. And it is the intersection between these two where we actually have our bistable solutions for the specific environmental dynamics that we are exploring. Okay. So the next thing is that we observe that in this case, noise is both functional and under selection. Again, we are going to look into more details here. We were thinking about how the population moves here, how we find the solution. And here I'm showing you for different environmental fluctuation frequencies, going from fast fluctuating environments to slow fluctuating environments, how this parameter, NH, that is a measurement of the ultrasensitivity in the system, changes for the different conditions and different mutation step sizes. This is specific for the control, in other words, deterministic gene expression. And what we observe is that it basically doesn't care about the environmental fluctuation frequency, it only cares about the mutation step size. That makes sense because it's basically residing in this area where the genetic potential is maximal, but it has to cross the bistable region because it doesn't have stochastic switching. So as the mutation step size becomes bigger, it can actually extend higher and still do an efficient transition. If it has a very small, it is restricted. So far, it makes sense. Now, if we look at the full system with the stochasticity, then we observe a very different behavior. In this case, if the environment fluctuates really fast, it is restricted to small values of the ultrasensitivity, and it starts moving away to higher values of ultrasensitivity as the fluctuation becomes smaller. And the parameter, the mutation step size doesn't seem to be as important here. So we wanted to understand what was happening. Again, we can go and look in a different direction for more details, and we are going to be looking at the adaptation dynamics. So what I'm going to show you here is first, what is the population fitness exactly one generation after each environmental change as a proxy for the adaptation time. And I'm going to show you the population fitness exactly 9 generations after an environmental change as a proxy for phenotypic robustness. The first thing that I want to highlight here is that there is a clear trade-off. You cannot optimize both of them. You either optimize the adaptation time or you optimize the phenotypic robustness. And it's very much the ultrasensitivity parameter that is related to the genotype, the one that would be defining which of these two are you prioritizing. And if we look at the distribution selected, of the population selected for different environmental fluctuation frequency, we observe that if the environment fluctuates really fast, it is prioritized a faster adaptation time. And if it fluctuates really slow, then we prioritize phenotypic robustness. This again makes sense because the relative cost of the adaptation time is going to be different depending on how long the population actually stays in the same system. Again, we are observing the adaptation to the environmental dynamics per se. Now, the adaptation time will depend on the genetic potential if we are adapting genetically, or on the epimutations if we are adapting through epigenetic switching. But both of these systems have to deal with the biochemical noise load, and that will directly affect the phenotypic robustness. So to better understand this, we have to look into the details of noise. Okay, so once again, we do this map because we know the system. Now, the noise load will depend on which of the two environments you are evaluating because it's very much related to the fitness. But basically, we overlap these two maps to get an information. If you consider the both environments, then it is actually optimal in terms of the noise load to be in the bistable region. This was somewhat new. People assume all the time that having an epigenetic switch will come with this cost for noise in general. But the reality is that when you take in account that noise is omnipresent anyway, it might actually be beneficial for controlling noise to be in the genetic region. And here it's hard to see, but basically the noise also reduces in the very center as you increase the ultrasensitivity parameter. Now, of course, if we are in the bistable region, we still want to do this epigenetic switching. So you have to consider that it might be problematic for you to go too deep into here, as it would take too long to actually have an epimutation and do the transition. So this explains the ultrasensitivity pattern that we are observing. Okay. Anyways, so
sorry about that. This is not something I can control. It's something by the institute.
Yeah, no, it's good.
Yeah.
The next thing I want to mention is that we actually use the lineages for reveal what is being selected here. Again, I would love to go into details. I don't have the time. It's a really beautiful story, but by now only believe me that if you look at the population in any given moment, so many things are happening. It is actually things that are adapted to the current environment that might not be informative. We wanted to understand what was happening in terms of the environmental fluctuation. And then the way to look at this is actually look at the lineages, look at the population that have survived a full environmental cycle. And then everything starts to fall in place. And we can actually catalog the different adaptation strategies in three. We have the epigenetic switching whenever the genotype is bistable and we observe no mutation at all in the lineages to be able to adapt. We observe as well genetic adaptation when the phenotype leaves the bistable region and we have at least 2 mutations to adapt in each direction for the environmental changes. And finally, we also observe this hybrid strategy where the population doesn't leave the bistable region, but we still have mutations that are basically biasing the transitions accordingly as environment changes. And finally, just to close, we observe that different evolutionary conditions favor different adaptation strategies and that epigenetic switching is favored exactly in fast fluctuating environments because it allows for fast adaptation while keeping the biochemical noise load small. Here's just showing you the 2 more relevant evolutionary parameters for us. It is the environmental fluctuation frequency and the mutation step size. And we observe that when the environment fluctuates really fast, epigenetic switching is always favored. If the environment fluctuates lower, then it depends. If you have a small mutation size, then you will choose this hybrid strategy of bistable adaptation, and otherwise you go to the traditional genetic adaptation. It is important to highlight that this is not an accident. Epigenetic switching is being selected because of the potential of stochastic switching. If we remove the noise, we don't see anything like that. Everything adapts genetically as observed. And as I was mentioning, this has been published actually for some time now. It has a lot more details about these technical parts, but it's very much the inspiration behind my research group. So we are continuing this idea by adding plasticity into the mix or the competition with oscillations or rhythms as another strategy to deal with fluctuating environments. And with this idea that we care about fluctuating environments in general, we also have this different avenue in the lab where we are looking at the strategies that now suppress the effect of the environmental fluctuations or noise by a phenomenon that is known as homeostasis of feedback control. But I didn't talk anything about that today. Okay, so with that, I finish. Thank you very much.